Mm. What do I want you to know? Where it occurs. I think I've said it 3 times now, right? They say in advertising, you got to say everything 3 times. So there's the 3rd time. Okay. How is that messenger RNA read? By attaching it to those ribosomes. And again, it's the tRNA molecule that brings those individual amino acids that are being read to the party to be read and form this string of amino acids. And that's what they're showing right here. All these blue arrows right here, they're all pointing to amino acids, proteins. All of these things are proteins right here, right? They look so individually pathetic. But all of these little amino acid strings right here code for something. Here's what these base pairs look like, or these triplet base pairs look like, guys. Remember, they're complementary, so TAC would be AUG. And there's methionine, that is your start codon. I'm not going to ask it, so I was going to start, but I'm not going to start. Okay. There's UAG, again, it's a stop codon, 1 start, 3 stops. All right, but that's just one of them. But you can kind of see all of our amino acids, okay? All of our 20 different amino acids, and there's only 20 of them. Okay. And here are some of these amino acids right here, and these are the DNA, and this is the RNA, complementary base paired with it, CGA, okay, AAA. Okay. So those are complementary base pairs that I was talking about. Right. But all of these messenger RNA, and there's my codon, my triplet of amino acids. Okay. This codon gets read, and the tRNA molecule brings methionine. We'll bring whatever the codon says to bring. Okay. So the tRNA molecule is bringing these individual amino acids together, or to the ribosome. There at the ribosome, magic happens, and we form a little peptide bond. And that's the peptide bond right there. Okay. And that's what connects all these individual amino acids into a string. And that string of amino acids is referred to as that primary structure. Okay. Just the individual amino acids. tRNA is what brings these things there. And here's what tRNA looks like. Okay. Here's what tRNA looks like. tRNA looks like. It looks like a T, I guess. So that's where they get the name. Okay. Okay. And on the tRNA molecule, about the only thing you really need to know is that the tRNA molecule has the anticodon to the codon. Okay. On a tRNA molecule, it has the anticodon, and that complementary base pairs with the codon. It docks with the codon, And on the opposite end of it, it'll have the specific amino acid. It'll be bringing it along. Okay. And it's that little amino acid. See, here's a nice little picture. Here's all my codes, my codon on the messenger RNA molecule. Here's tRNA. Here's the anticodon. Okay, so there's the codon, and there's the anticodon. Okay. And there's the amino acid that specific tRNA molecule is bringing to the party. Brings it in, forms a little peptide bond here. That's what that arrow is trying to show you. There it is. Okay. We move them down. This thing just slides on through. So these guys will slide over a spot. Here's the ejector seat. So that tRNA molecule gets kicked out. Got an open spot, accepts the next tRNA molecule, and we form a little peptide bond again, and we'll just keep moving along until we reach a stop codon. Okay, until we reach a stop codon. This is happening all over the place, wherever I have ribosomes involved. Again, they could be floating out within the cytoplasm, or they can be attached to the endoplasmic reticulum. So there's my string of amino acids being strung together there, and they're just stuck within the cisternae of the rough ER. And we'll fold it, we'll manipulate it a little bit to get this thing ready for action. And this is just looking at an example of a kind of secondary structure here. Okay. As this long chain of amino acids gets longer and longer and longer, it'll start to bend and fold on itself, right? And twist. When it does that, we start getting all these crazy bonds. So it can maintain that secondary shape. If we have a number of these secondary structures here coming into contact with one another, they can form a tertiary. We get a number of tertiary structures interacting with one another, we get a quaternary structure. And we talked about that last week, I believe. But that primary sequence of amino acids, that primary structure, that gets built at that ribosome, at that rough ER. Okay. And depending upon what we're making, depends upon how far that goes in its complexity and in its shape. From the rough ER, where do they go? We talked about it just a handful of minutes ago. It goes to the Golgi complex. There at the Golgi complex, it's going to be further modified. Okay. Package up in a little bitty vesicle. And then it goes to wherever in the world it's supposed to go. Okay. Goes to wherever it's going to go. Sometimes it doesn't go that far at all. Sometimes it's packaged up in a little bitty vesicle, travels a little bit further within the cytoplasm, and a lysosome will bind with it. Break it down. And we'll utilize those individual amino acids to make other things within the cell. In this video that I'll pull up here in a second, cells recycle pretty much everything that they can. And when it comes to proteins, proteins get reused and reused and reused over and over again as long as they stay within that cell. They help to make that cytoskeletal structure that we mentioned earlier. Okay. I don't care about ubiquitin. Okay. Ubiquitin is just a marker that says, hey, go kill that guy because I want to use his stuff. Okay.